Welcome back to the deep dive. Today we're digging into some fascinating ecological detective work. We're looking at a massive kind of silent invasion. The African clawed frog isn't surviving where it shouldn't be. It's actually thriving across the French-Belgian border. And this is in conditions where, well, climate models basically said it couldn't last long term.
Yeah, that paradox is really what makes this research so compelling. We're analyzing the findings from the paper, mapping the northernmost transnational non-native population of Xenopus laevis using pooled eDNA sampling, and the lead author is Everts.
And it's critical work, isn't it? Because globally, freshwater biodiversity is just plummeting. And these
Right.
non-native invasive species, NIS, they're a huge driver of extinctions and ecosystem chaos. We desperately need better ways to spot them early and react fast. The EDRR systems, early detection, rapid response.
Absolutely, and the specific culprit here, Xenopus laevis, the African clawed frog. It's native to sub-Saharan Africa, obviously, but it's been used so much in research labs, pregnancy tests back in the day, even the pet trade, sadly. So now it's got populations established globally, North, South America, Asia, Europe. It's everywhere.
Okay, let's unpack why this particular frog is such a successful invader. It sounds pretty bad. It's highly fertile, almost totally aquatic, and really cryptic, meaning it hides exceptionally well. So its arrival can go unnoticed for years, sometimes decades. But once it's established, X. laevis just hammers native wildlife. Predation, competition, even spreading diseases. And it moves. Apparently it can disperse up to 5 kilometers a year. It doesn't just stay in one pond.
Exactly. And this specific population, the one straddling the French-Belgian border, it's the northernmost one known.
Yeah.
Trying to monitor this thing traditionally, it's a nightmare. You've got this huge area, 3 different administrative regions involved, and the usual methods, netting, visual surveys, they're slow, costly, and honestly, pretty ineffective for a creature this secretive.
Right, which is where eDNA comes in, environmental DNA. This has really shifted the game for ecological monitoring. So for you listening, eDNA is essentially the genetic breadcrumbs, skin cells, mucus, waste that creatures shed into their environment, like water. You take a water sample, filter it, extract the DNA. It's often way more sensitive and cost-effective than trying to physically find the animal, especially for, rare or elusive things in ponds or streams.
Mm-hmm. And the big innovation in this study wasn't just using eDNA, it was how they sampled it. They needed to cover this vast area, 426 sites in total, but they had to slash the lab analysis costs somehow.
Wow.
So they developed this strategy, pooled eDNA sampling across locations.
Pooled sampling. Okay, that sounds efficient. But how does it actually work on the ground? Are they just mixing water bottles together?
Sort of, yeah.
Yeah.
Essentially, they grouped nearby sites into clusters. So instead of one sample per pond, they collected water from several adjacent sites, usually between 2 and 8, about 4 or 5 on average. Then they merged all that water and filtered it through just one eDNA filter. This meant they could cover that huge area by analyzing only 83 of these pooled samples, plus of individual ones for validation. Much cheaper.
Okay, a high throughput approach. And then for the analysis, they used quantitative droplet digital PCR, ddPCR. You hear absolute quantification. What does that mean in practice? Are they literally counting DNA molecules?
Pretty much, yeah. That's the beauty of ddPCR. It takes the sample and divides it into thousands, maybe tens of thousands, of tiny droplets. Think of it like running thousands of tiny PCR reactions all at once in parallel. By counting how many droplets light up positive for the target DNA, you get a direct count of the molecules. No need for standard curves like with qPCR. It gives you incredible precision, even in mucky pond water that might have stuff that inhibits other PCR methods.
Okay, so super efficient sampling, super precise analysis. But I have to ask, if you're pooling water from, say, 8 sites, isn't there a big risk that if the frog is only in one pond at really low density, its DNA just gets diluted too much in the pooled sample, leading to a false negative? How do you balance that cost saving against the risk of missing something important?
That's the crucial question, absolutely. And that's why they did specific validation tests. They took 2 clusters, each with 4 ponds, where the initial pooled sample came back positive for the frog. Then they went back and sampled each of those 4 ponds individually. And interestingly, in both cases, the frog DNA was actually only found in 1 out of the 4 ponds in each cluster.
Ah, so the pooled sample still picked it up, even when it was only present in 25% of the sites contributing water.
Exactly. Even at what were likely low densities in those single ponds, the pooled method still worked. It detected the frog clearly. That was a big relief.
Okay, that's a huge win for efficiency then. Test 1 pooled sample, effectively screen 4 locations.
Right. And they also checked the quantification. In a cluster where the frog DNA was more concentrated, the The pooled sample gave a concentration of 1.02 copies per microliter. The average concentration across the 4 individual ponds was 1.05 copies per microliter. That's incredibly close, like 97.5% similar. Now, admittedly, the variability went up when concentrations were really low, but for the main goal broad area mapping, figuring out presence-absence quickly, it proved really successful.
So, okay, they validated this clever cost-saving method, testing multiple sites for the price of one, essentially. What did the main survey, the big one spanning 2020 to 2023, actually find? Did this efficiency pay off in revealing the true scale of the invasion?
Oh, it paid off all right. The scale they uncovered is genuinely alarming. Xenopus laevis DNA was detected in 26 samples overall, 15 pooled, 11 individual. And when they mapped these positive detections, the total occupied area came out to a staggering 103 square kilometers.
103. Wow, that's much bigger than I would have guessed.
It's huge. And the spatial analysis showed it wasn't just scattered hotspots. It formed a single continuous area. This suggests one large, interconnected population stretching across parts of France, and both the Walloon and Flemish regions of Belgium.
So not just isolated outbreaks, but one continuous population crossing regional and even national borders over 100 square kilometers. That is way beyond what people thought was happening there. How is this aquatic frog managing to spread over such a massive area? Did they find any clues?
Yeah, the data points very strongly towards specific waterways acting as dispersal corridors. Every single positive detection was within 1 kilometer of either the main river, the Lille Grijs, or its smaller tributary, the Doorn stream.
Okay, and what's really fascinating here, as you mentioned at the start, is how these findings just smash previous assumptions about this frog.
Totally. This French-Belgian border region, it's significantly colder than where most established Xenopus populations are found around the world. People thought the climate there was basically unsuitable for the frog to establish long-term. So finding a thriving, huge population there challenges our models of climate suitability and thermal tolerance. This frog is clearly tougher, or adapting faster, than we assumed. It really makes you rethink where else in Northern Europe it might be able to establish.
And covering 103 square kilometers, that doesn't sound like an early invasion anymore, does it?
Not at all. It's clearly way past the early detection phase. That huge area strongly suggests the initial introduction probably happened years, maybe even decades ago. The first official record in Belgium was only 2006, which implies it could have been introduced almost 20 years ago, or even earlier. Because it's so cryptic and wasn't systematically monitored across borders, it just spread silently. The paper says the window for complete eradication has narrowed considerably.
Okay, let's zoom in on those dispersal corridors you mentioned, the River Lies and the Doorn stream. Was one more important than the other?
Definitely. The Doorn stream showed way more positive detections and significantly higher eDNA concentrations compared to the Lys. The Lys is a bigger river, yes, but it's heavily engineered, canalized, steep banks, fast flow, not much good surrounding habitat for the frogs.
So the smaller stream, the Doorn, is acting like a kind of frog superhighway.
Pretty much, yeah. It has more natural vegetated banks, and crucially, it flows through areas with lots of artificial ponds, often linked to agriculture. These ponds tend to be turbid, eutrophic meaning full of nutrients, probably from farm runoff. And Xenopus seems to love these kinds of conditions for breeding and thriving. The study found really high eDNA concentrations, which usually points to breeding activity right near the source of the Doorn.
And if we connect that to the bigger picture, high concentrations, breeding activity near the source, what's the major ecological risk there?
The big risk is further spread into connected river systems. Finding high concentrations near the Doorn source puts the ecologically valuable Yser catchment directly downstream at risk. The Yser is important because it holds remnant populations of native amphibians, like the great crested newt, which is protected. And unfortunately, those native newts and frogs are really vulnerable to Xenopus. It eats them, competes with them. So an invasion into the Yser could be devastating.
Okay, so we have this massive, confirmed transboundary population, clear dispersal routes identified, and a significant downstream threat. This raises a really important question. Given that Xenopus laevis is listed as a species of EU concern, which mandates action, what is the management response?
Well, that's probably the most sobering part of the paper. Despite the clear scientific evidence, despite recommendations, despite the EU regulations requiring coordinated action, the study states that no explicit management decisions have actually been implemented in any of the 3 affected regions, France, Flanders, or Wallonia.
Seriously. So the technology, the pooled eDNA, delivered this incredibly detailed, timely, actionable map of the invasion. But the administrative follow-through, the coordination across borders just isn't there yet. If that window for affordable eradication has narrowed considerably, as they say, are we basically watching an intervention that could have been manageable turn into a permanent, expensive, long-term control problem because of this delay?
That appears to be exactly the risk. The science effectively closed the knowledge gap. The pooled eDNA method worked, it was cost-effective, it provided the map. But the policy and coordination gap remains wide open. The paper really stresses the need to move past fragmented, localized efforts. They're calling for standardized, harmonized monitoring and management right across the borders, underpinned by, actually sharing the data effectively between France and the Belgian regions. It's become more of a political and administrative hurdle now than a scientific one.
And assuming that coordination does eventually happen, this detailed eDNA data could then guide very specific actions.
Absolutely. You could use the maps to target eradication efforts much more precisely. For example, using quicklime treatments, which can kill the frogs, but only in those specific ponds identified as heavily infested breeding sites, or strategically placing physical barriers to block dispersal along key stretches of the Doorbeek. And of course, using eDNA for ongoing monitoring to see if the interventions are working. The data makes truly adaptive management possible, but only if the different authorities agree to act together.
Okay, let's summarize the key takeaways from this deep dive then. We've learned the northernmost Xenopus invasion is way bigger and more established than anyone realized. It's thriving in a climate thought to be unsuitable, and it's spreading mainly via specific human-altered waterways, especially the Doorbeek. And crucially, this whole situation was mapped out efficiently using this novel pooled eDNA sampling strategy.
Yeah, and I think the core insight, maybe the slightly provocative thought for you, the listener, is this. We have this amazing technology now, like eDNA, that can map these complex biological invasions across huge areas, even across countries, really rapidly and accurately. But that knowledge is only truly valuable if it's matched by effective centralized coordination and data sharing between the responsible authorities. And maybe, considering this frog readily uses those linked agricultural ponds, maybe future surveillance shouldn't focus so much on the lines on a map of the national borders, but more on breaking the physical connections between those human-altered habitats that let the invader spread.
That's a really interesting point, shifting focus from political boundaries to ecological bottlenecks. Definitely something for policymakers and ecologists to chew on. It highlights the complex challenges of managing these transboundary invasive species in our interconnected world. We encourage you to keep exploring this critical issue.